Dr. Borad is going to be our first speaker for the session. Mitesh, a good friend of ours, and is also one of the leading figures in cholangiocarcinoma and the treatment of cholangiocarcinoma. He's also one of the most innovative clinician scientists. Mitesh actually is one of these unique physicians that essentially spends a lot of time at every interface of what we do. He's a basic scientist and brilliant at doing that. He's a translational scientist. He knows how to bring things from the lab to the clinic, and he's also one of the best clinicians I've worked with and the best, most compassionate physicians in taking care of cholangiocarcinoma patients. Without further ado, it gives me great pleasure to introduce my great colleague who's going to give us an overview of innovative biomarker trial strategies in biliary cancer.
Okay, thank you, Tony. That was very kind. Even my parents wouldn't be able to give such a warm introduction. So I'm following a very exhilarating talk that was challenging, how we approach things. And I thought I'd make this talk relevant with just a few simple messages. It's getting late in the day. Everybody's been very attentive and present at all the sessions, so I don't want to make it any more difficult to get through the last few hours than it needs to be. So while we'll go over trial strategies here, I won't bore you with details of genetics and these types of things. I'll just give you some concepts and how maybe we should challenge them as a patient, researcher, and industrial community. Okay, so the Brits have a way with words, I must say. I use dogma as my central message, so defeating dogma is what we need to do. And Webster didn't have a good definition in the American dictionary, so I used the Oxford English Dictionary definition. So it's a principle or set of principles laid down by an authority. In this case, the authority is ourselves in a way, as incontrovertibly true, which basically means just do it this way because I said so. And once you fall into this trap, it's all downhill from there. Okay, so a lot of you have heard through the last few days about getting genetic profiling done. And then when you hear about this, you hear your doctor say, like, okay, let's see if you have some tissue. If you don't, let me get a biopsy, and then we'll wait, 3-4 weeks before the results come back, and then we'll talk about it, right? It makes sense in general because that's where the cancer is, and that's where you're going to find the highest yield if you go and get the sample that has the most concentrated material. However, I feel that this is a dogma, and that as you may have heard in Dr. Corcoran's talk and other talks through the day, this will probably not be the way we assess mutations in patients as the first strategy. So tissue or blood or both. So right now, the answer might be more, well, tissue or both. I don't know about blood. That sounds not reliable. It's not standard of care, right? These are all sort of dogmatic terms that are used, but if you think about where the technology already is and where we're going, this will probably be the standard of care. So why not accelerate to get to that standard of care faster than build our own resistance to the idea, right? So you've heard of liquid biopsies being used for different things, detecting the disease early, say in high-risk patients. If anyone here has been told they had primary sclerosing cholangitis, that's one of those sort of high-risk conditions. Could you use a blood test to maybe find the cancer early? When you want to do the mutational profiling, instead of running straight to the tissue, wouldn't it be great if you could just analyze the blood sample and get the result in a few days? And then if you don't find anything, maybe then go to the tissue. Every few months, patients who are getting treatments get scans. And that's sort of the standard of care way of measuring if the treatment's working or not. Maybe these types of assays will replace the scan, so you don't at least have to have them as frequently. And then, of course, the tumor is sort of like a living body in a way that evolves. So if you need to do some type of test on a frequent basis, you can use a blood test. It's easier to do and then do it repeatedly. Dr. Goyal and her colleagues at Mass General have highlighted many of the aspects that I showed here. You can see that this patient that was treated with a category of drugs called FGFR inhibitors, which hit a specific target, yielded a response in the middle scan. And then after a while, it started getting worse. And you can see that the same conclusions could have been made just from the blood test if you had them available to do rapidly. Maybe you would not have needed the scans per se to make these assessments. We're just used to doing them. Additionally, these tests were also able to tell why the cancer had become resistant. So in a way, they added even more value than the scans. So prediction. Likely this type of testing will become the preferred, not the replacement method, but the preferred method, maybe the one we'll go to as a first start, not really just go to the biopsy. Right now we're doing this assessment at different time points, only sort of in clinical trials. Not everybody's doing it. People are on the fence. Some are believers, some are not. But I feel as the data builds up, the dogma will disappear. Currently, we're having to send all our tests to sort of labs. We're shipping samples, they're processing them, then they're sending us information back, take a look at the results. We might be able to do this in the local labs of hospitals as the technologies become better, so that it's faster, you're not moving samples around. And when we talk about, did the patient have a response right now, the sort of standard approach is use RECIST, R-E-C-I-S-T. This is use the scans. That's what the language the FDA understands. If we want them to understand a different language, maybe we need to propose different ways of doing the assessments. And if we do this as a group, and if this is reliable or more reliable, then this might be the way to do things. Okay. Another theme I hear in the conference a lot is what drugs are FDA approved for use in cholangiocarcinoma? Anybody know the answer? Zero? Okay. So I'd like to introduce a concept that's been around a while, but one of my mentors at TGen, Dr. Dan Von Hoff, when I was a fellow there, came up with the concept of approval by target. Will a drug get approved not for a disease, but as Vivek Wadhwa was mentioning, it's not about the disease, it's about the target, it's about the alteration, right? And people thought he was nuts, right? It took 12 years or so for him to get rid of that tag, but instead of cancers being labeled by organ site, et cetera, maybe they'll just be labeled by specific targets. So here, could you take the same target and study it in different cancers? And if you did it this way, could you subvert the whole process of sort of approval by cancer and go to approval by target? So one such drug is PEMBROLIZUMAB, which is an immune checkpoint inhibitor. And as everyone here knows and has heard about it for a few years, this has been shown to be effective in a wide variety of cancers. You can see a whole list there in that pie chart. And it works quite well in most of these cancers. And this study that was published in Science by Dr. Lay and colleagues from Hopkins was done in microsatellite unstable cancers, or MSI-HIGH cancers, not for any specific one, but just for this target. Similarly, there has been work with a category of drugs called TRK inhibitors. And again, this was an approach taken using approval by target, and you can see that there were a couple of cholangiocarcinoma patients in that list. One of them did have a response, and you can see the waterfall plot comprising of lots of different types of tumors by organ, but they all have the same underlying connection, which is gene fusions of the TRK pathway, NTRK1, 2, and 3, and most of the patients had some benefit by what you see as the downward decrements in the waterfall plot. So both of these drugs were approved for cancers for specific targets, and of course, you can use them for cholangiocarcinoma if you have a patient with this alteration. So the answer is maybe not zero. You can qualify it and say, yeah, there's 2 drugs already, and maybe more to come. Okay, so that was the sort of second dogma, and I'll contradict myself as I go through the third aspect here. Currently, the approach we've taken for getting drugs widely used, getting them FDA approved or getting the evidence that's convincing enough to everybody, has been centered mostly around drug companies or researchers. We say we have the patient at the center, but only momentarily, and then when people start designing trials, they forget all about it. So for an individual patient, what is the best approach? So if you look at the n=1 category, you can see different patients have different attributes, blue shirt and red pants, or a green cap, if you look at it from the clothing perspective. But none of those 6 patients have exactly the same makeup, and that would mean that each one of those patients would require an individualized approach. However, when we're studying drugs, again, because this is in the paradigm of interests of industry and researchers, we end up taking sort of a drug-based approach, and we say, well, let's take the 3 folks with the red pants there and study them, and then if they all kind of do well, then now you can use that drug eventually in n=1 as needed, right? But most of our efforts are going into drug-based approach. These 2 things seem paradoxical, like, they seem like oil and water. Maybe they don't mix. Can we combine these features and use the best of both worlds and make this patient-centric, right? So basket trials, which was the standard approach, and n=1. Can we combine these 2? We went over this earlier, so I won't spend too much time on it, but folks have come together, and this includes the various United States federal government-funded cooperative groups, also NCI Canada, the patient advocacy teams, and hopefully industry partners to define a platform that we've called BATCH, Biliary Cancer Alterations, Predict Therapy Choice. There's a large number of agents that could be applied to this because this is a very target-rich cancer, and many of these things occur at a low frequency. So in a rare disease at a low frequency, unless we take this kind of approach, we'll never be able to study these in sufficient depth, and they'll never become available widely so that everybody anywhere can use them in the n=1 way. So the proposed structure is we would do mutations matter at the initial diagnosis, hopefully using either tissue or liquid biopsy approaches. And by the time we need another therapy, the patients will have many options to choose from if there's a match in terms of a target. And even if there's not, there will be a category where patients can be treated on this trial with drugs that may not utilize these match-based approaches such as immunotherapy. So almost 100% probability of receiving something in the end. As I described before, this will require all the stakeholders to come together. This will be driven by patients, not by other interests, which should only be helping and not coming in the way. We talked about both tumor and liquid biopsies as dogma 1. And this will have full engagement of all the parties at the table, particularly patients who have been typically missing from the conversation. This, of course, requires a huge team. I want to thank all my colleagues, Dr. Goff and O'Dwyer, Dr. Saab, Dr. Al-Kuwari, Dr. Javli, Dr. Knox and Abu Alfa, folks from regulatory agencies, Dr. Merrick Munstrom, and most importantly, the patient community for supporting this effort. And Stacy and Kathy have been very instrumental in this regards. Okay, so the equation now is n equals 1 plus basket equals batch. So that's an easy one to remember. Almost 100% chance of receiving something if it makes sense. And then hopefully this will change the paradigm on how we study drugs to help patients in this disease. So I'll leave you with this last thought. Every dogma has its day. If you see one, just defeat it. It's not my own words. It's Anthony Burgess. And I'll conclude there and thank you for your attention.
All right.